Alright guys, so we're moving on and we're going to cover a little bit more about genetics, and we're going to take a look at some traits that pertain to humans, and we're going to take a look at some other types of traits and how they're inherited. And so just a little bit about our chromosomes again. So hopefully at this point we know that we have 46 chromosomes in a normal human, or 23 pairs. Well, how do we actually know that? And we know that because we can physically take a picture and create an image or a little kind of a diagram of our chromosomes called a karyotype. And so remember that in most of the cell cycle, most of a cell's life, the DNA is in the form of chromatin, and it's lesser condensed and we can't really see it. But starting in mitosis, one of the stages or one of the events that happens in prophase is, remember that the chromosomes condense, and so we can see, like physically see, the chunky chromosomes with a really good microscope, not really with the ones that we use, but in a good microscope they can actually see the chunky chromosomes throughout the stages of mitosis. And so starting in prophase, we get those condensed chromosomes, and we have chromosomes all the way until we get to telophase, and then it starts to go back to that chromatin form. And so in the earlier stages before metaphase, they would be duplicated. Remember, they'd be X's. If you took the picture after they were separated in anaphase, then you would have single chromatids. But either way, at some point in mitosis, they take pictures, and then they're obviously not going to be organized. And so what they do is they kind of blow the pictures up, and then they do a cut and paste job. And later this week, you guys are going to do an activity that kind of mimics doing a karyotype. And what it does is it creates this picture of an organism's chromosomes. Now, when we do this, we'll see this more, but they're organized by their homologous pairs. You can see that they're arranged from 1 all the way down to pair 23. Remember, they come in pairs. So this would be a homologous pair, that would be a homologous pair. And the way they can tell that they're homologs is they're normally the same size. So even though some of them are crooked, they're the same size. And you can see that the biggest pair is first, all the way down to your smaller pairs. And then they also have the same banding patterns. And so between the two, they're able to pair up the chromosomes. And so by doing this, by looking at a karyotype, number one, you can determine the sex of an individual, because pair 23, as we can see down here, are our sex chromosomes. And so either the individual has 2 X chromosomes, and if that's the case, they're a female, or they have an X and a Y chromosome, and if that's the case, they're a male. So one thing we can determine is whether or not they are male or female from the karyotype. But really the main reason we would do it is if they suspected a genetic disorder of some type. And so there's other ways they can determine if the baby's male or female. And so they may do a karyotype if they are, or they have some kind of evidence or some kind of clues that the baby might have a chromosomal disorder. And so we would use a karyotype to maybe identify aneuploidy, which is where they have an abnormal chromosome number. Maybe they have 45 instead of 46, or maybe they have an extra chromosome and so they have 47. They can also look and see if there's extra or missing pieces of chromosomes. Like for instance, there's a disorder called cri-du-chat, and it means cry of the cat. But in that particular disorder, there's an extra piece on chromosome 5. Or maybe there's a missing piece on chromosome 5, and so the chromosome would not look completely normal. Now, we can also look at whether the chromosomes affected are either the sex chromosomes or if they are autosomes. And so pairs 1 through 22, we need to understand the difference here, because when we look at inheritance of traits, one of the things we're going to determine is it is an autosomal trait or is it a sex-linked trait. And that depends on what chromosomes the genes are on. And so autosomes are your pairs 1 through 22, and I'm going to go back for a second. And I want you to see that on chromosome pairs 1 through 22, everybody, okay, this is a male right here. You can see the XY. And this one down here is a female. You can see the 2 X's. But on pairs 1 through 22, everybody should have 2 of the same chromosome, okay? And so when it comes to autosomes, Everyone should be getting one from mom and one from dad. Those are our homologous pairs. And then on the sex chromosomes, pair 23, you can see that not everyone is going to get 2 copies of the same chromosome. A girl would get 2 copies of the X, and a boy will get a Y instead of a second X. And we'll see more on that later. So some of the first genes to be discovered for humans were our blood typing genes. And there's actually multiple components to our blood type. And that's why, like, if you were doing like an organ transplant, you need to have more in common than just the same blood type. But in terms of our blood type, we typically look at 2 blood groups. We have the ABO, and then we have the RH blood group. And so if you know what your blood type is, it may be something like O positive or A negative. And so the first part of your blood type, the letter or letters, that is the ABO blood group. And then the second part of your blood type, the positive or negative, that is the other component, the RH blood group. And so we're going to start by taking a look at the ABO blood group. Now, this was one that was an example of multiple alleles. So we actually have one gene that determines our ABO blood type, but instead of just 2 possible alleles, we have 3. We have the A allele and the B allele, okay, and they are both dominant, they are codominant. Now, when you're doing your Punnett squares, it doesn't matter what you use, but technically these are represented by a capital I with a capital A or a capital B superscript. And because this indicates that they are both dominant, okay? So the capital I comes from the word immunoglobulin, which has to do with the protein. Now, when you're doing your Punnett squares in the next couple of days, you can use capital A, capital B. You can use the I's. It doesn't matter as long as you understand the blood types, okay? So we have 2 alleles that are dominant, A and B, and then the O allele is recessive. And because it's recessive, it's represented by a lowercase letter, the lowercase I. Once again, it doesn't matter which one you use as long as you know that O is recessive. Now, what this does, what these genes do, is they determine what antigens are on your blood cells. And an antigen is just a protein. There's antigens on all of your cells. And so antigens are proteins that are found on the cells, and antigens are proteins that when they're not recognized, they will lead to the production of antibodies. Now, antibodies are part of our immune system. So when something enters your body and you don't recognize it, what that will do is that foreign antigen will trigger the production of antibodies. It's part of our immune response. So we don't necessarily, in this case, want to produce antibodies. But this is why we have to be careful about what kind of blood you receive or what kind of blood you give to a person, is because we need to have our antigens and our antibodies straight, okay? So the way it works is if you have the A allele, okay, then you are going to have the A antigen, which means you have this particular protein on your blood. If you have the B allele, then you're going to have the B antigen. So if you get both of them, AB, you have both of those antigens. Now, O is recessive, and so what happens is O doesn't get the A antigen or the B antigen. And so that is going to relate to what antibodies are produced. So you produce antibodies to what you don't have, okay? So if you're type A, you notice you have the A antigen, but you would produce antibodies to what you don't have, so antibodies to B. If you're type B, you have the B antigen, and you would produce antibodies to A, because you would not recognize those. If you're AB, that means you have both antigens, and therefore you would have no antibodies because you have both of the proteins. And then if you were group O, you don't have any of the antigens, so you produce antibodies to both. And so this right here is why you can't just give blood to anybody, because you don't want to give someone an antigen that they do not recognize. Now, before we talk about the donors and receivers, we're going to add the other blood group in, and that's the RH blood group. Now, the RH comes from the fact that it was discovered in the rhesus monkey. That's really the only significance to the RH. And once again, we have a single gene, but this time there's only 2 alleles. So there's positive, which is dominant, or there's negative, which is recessive. Now, if you were doing a Punnett square for this, you could use, like, because it's the D antigen down here, you could use like a capital D to represent the positive allele, and you would use a lowercase d to represent the recessive allele. And you would work this out just like any little monohybrid Punnett square. You could be homozygous to be positive, or you could be heterozygous to be positive. The only way you would be negative is if you got 2 recessive alleles, okay? Now, just like before, if you're positive, it just means you have a protein or an antigen on your cells. In this case, it's called the D antigen. And if you're negative, you would not have the protein on your cells. So when it comes to donating and receiving, you know, we do not want antigen and antibodies meeting, because what happens is the whole purpose of an antibody is made to attach to an antigen. And when that does that, it can cause your blood to clump up or agglutinate. And that does not sound like a good thing. So this is why if you had a type A person and you gave them type B blood, they're not going to recognize that B antigen. So they're going to start producing antibodies, and then that will cause their blood to clump up. So it's really important that we give like a compatible blood type. Now this is where we get into the universal donors and the receivers. Now O did not have antigens, and so it doesn't have these proteins that other cells can recognize as foreign, and so we call the type O person the universal donor. Now I do want to be more specific and say that it's really O negative, because you cannot give a negative person positive blood. Okay, so if we include the Rh factor, O negative is the true universal donor. So if you have O negative blood, I guarantee you they're going to want your blood. They're going to want you to donate, because they could give that to pretty much anyone. Okay, now if you're O positive, they still want your blood, because that's the most common blood type. And of course, they could give that to anybody that had a positive type of Rh factor. Now, AB, on the other hand, because they have both antigens, they don't produce the antibodies, which means you can essentially give any blood type to an AB person. And once again, we're going to include that Rh factor to be more specific. So the true universal receiver would be AB positive, because they have all the antigens. They have the A, the B, and the D antigen. And so that's how you kind of coordinate with the blood typing and who can get what. So if you're type A, you can get A or O. If you're type B, you can get B or O. If you're AB, you can get any blood type. And if you're O, here's the kicker with O, you can only receive O, okay? Now, if you throw in the Rh factor, a positive person could receive positive or negative blood, but a negative person can only receive negative blood. All right, so now we're going to move on to like sex chromosomes and sex linkage. And really our focus is about humans, but I just kind of wanted to give you a little bit of background info that normally it's the chromosomes that determine the sex of an organism. And so what we have is that XY system, and this is really the only one that we're going to worry about, the only one you really have to know about. But I do want you to see that not all animals and not all organisms have the same thing. So like for a lot of your insects, an XX would be a female, but just a single X would be a male. Some animals have this ZW system, where males are 2 Z's and females are ZW. For some, there's a difference in the number of chromosome sets. So you can see that like in ants and bees, females have a diploid chromosome number, males only have a haploid. And then for some animals, it has to do the temperature at which they incubate when they're in their eggs. Different temperatures would lead to the expression of either female genes or male genes. So really, once again, all we need to focus on is the XY system, because this is the one that pertains to us. But here's just to show you just some examples of other organisms, how they determine their sex. All right, so that brings us to sex-linked genes. And so just like the name implies, the sex-linked genes are genes that are on the sex chromosomes. Now, if you look at pictures, what you'll find is that the X chromosome is a pretty normal-sized chromosome. It's got lots of different genes on it. But the Y chromosome is a little bitty tiny chromosome, okay? So normally when we're talking about sex-linked traits, typically they are going to be X-linked, which means they are genes that are carried on the X chromosome. Now this is going to be a little bit different than autosomal traits because as you can see, a girl will get 2 copies, but a boy will only get 1. And so that's going to affect the inheritance between males and females. It's going to be a little bit different than in an autosomal trait. So we do have a few different examples of some genetic disorders when you do your Punnett squares. These are all 3 examples of X-linked traits that are recessive. And so all 3 of these are going to be more common in boys than they are girls. And we'll see why as we get to pedigrees. And a little bit more on the Y chromosome. So remember, it's tiny. In comparison to the X, it's little bitty. And obviously females do not have one. So it can't have any information that is absolutely essential to being a human. Now it might have some information that's essential to being a male, but it does not have essential information to be a human being because women do not get a copy of the Y chromosome. There's not a lot of genes on there. There's one in particular that's responsible for having the male parts. There's others that are responsible for sperm production. But like I mentioned, for the most part, your Y chromosome is going to give a maleness to the individual. And then the females don't have one, so it can't have critical information on it. Now, when we talk about your homologs crossing over, like in meiosis, because we have 2 different chromosomes here, we're not going to see crossing over between the X and the Y. And so essentially that Y chromosome is going to be passed from father to son, and then down to the grandson, and then down to the great-grandson. For the most part, it's going to remain unchanged. And so this does make it easy to kind of trace the lines through fathers to sons and down further generations. All right, and that's going to take us to pedigrees. Now, kind of like a karyotype is a picture of our chromosomes. It's a way for us to study, different disorders. Pedigrees are another type of diagram that can help us trace a particular trait through a family. Now, you have to understand that any trait could be traced. You could go home today and you could create a little family tree and you could pick any trait that you wanted. Like I have very curly hair. So if I wanted to, I could go through and I could look at my parents and my grandparents and I can look at my children and I could create a pedigree of who all has curly hair in my family. It doesn't matter whether it's a dominant trait or a recessive trait, an autosomal trait or a sex-linked trait. A pedigree can trace any trait, okay? Now, once you have this pedigree, we can analyze them and we can kind of look at an inheritance pattern of how is this trait passed on from one person to another. And so in order to create a pedigree, we do need symbols. So a circle is always a female, and if that confuses you, I always remember that females have curves, okay? And then squares are males. And so if you have a non-filled in, that means they do not have whatever trait that we're tracing, okay? If they are completely filled in, that means that they do have whatever the trait that we're tracing is. So if I was tracing curly hair and I had a dark circle in my pedigree, that would be a female with curly hair. If I had a male that had straight hair, then he would be a square that is unfilled. Now, sometimes in pedigrees, you'll see carriers. And a carrier is a heterozygous individual, okay? But it's really important to understand that carriers only exist if the trait is recessive, okay? Because a carrier is a heterozygote. Now, by definition, a carrier is someone that carries the recessive allele, but obviously they would not have that recessive trait. So carriers are only seen if the trait is recessive. Because if I have a heterozygous individual and the trait is dominant, they're going to be a completely filled in circle or square, okay? So carriers are heterozygotes when the trait is recessive. Now, also in pedigrees, we see relationships. So if you see a horizontal line between a circle and a square, that means a marriage or a mating, okay? They don't actually have to be married. And then a vertical line comes down and it branches to all of their children. So you can see that this is a female with the trait. This is a male without the trait. So they produced 1, 2, 3 children, okay? They have a son that's a carrier, which means he's heterozygous, so this trait must be recessive. They have a daughter that does not have the trait, and then they have another son that does have the trait being traced. Now, these lines go out to their spouses or their mates. So this son, this would be their daughter-in-law or their soon-to-be daughter-in-law maybe. And then this would be another daughter-in-law. And then they have a child, and that's how it goes. So we're going to look at kind of what do we do with the pedigrees. And you actually have some practice in your packet, and you have some questions that you need to answer about pedigrees in your homework packet. And so if you're studying a pedigree, normally you're going to have to answer questions about two things. Number one, how is it inherited? And so in terms of inheritance, you can identify whether it's a dominant or recessive trait, whether it's autosomal or sex-linked, okay? And so if I'm trying to determine the inheritance pattern, if it's a dominant trait, remember it only takes one dominant allele. If it's a recessive trait, then the person must have two of the same alleles, okay? And so we can kind of look at a trait and try to determine dominant or recessive. And then another thing we need to determine is it autosomal or sex-linked? Now, remember that if it's autosomal, everyone's getting 2 copies of that gene. And so an autosomal trait would not show a significant difference between the males and the females either having the trait or not having the trait or being carriers, okay? So if you don't notice anything really different between the circles and squares, you have about an equal number of, those that have it and those that don't, you don't really see anything majorly different between the circles and the squares, then that's most likely going to tell you that it's an autosomal trait. If it's sex-linked, you will see a difference in the inheritance pattern. Okay, now remember that most sex-linked traits are X-linked, and a lot of your X-linked traits are recessive. Now, girls get 2 copies and boys only get 1. So the thing with males is that it only takes 1 recessive allele to show the trait. Normally it would take 2. So girls get 1 that can cover it up, but boys only get 1 because they're getting a Y instead of a second X. So if it's an X-linked recessive trait, boys are going to have it more. Another difference is that if it's X-linked recessive, only girls can be carriers. Boys are not heterozygous because they get an X and a Y. Okay. Now, if it's Y-linked, a Y-linked trait, that would only affect the males because females aren't getting 1. So normally when you see sex-linked, you see X-linked recessive. So here's just a few different examples of some pedigrees. Whenever you're doing your pedigree work, you can come back to these and they may be helpful to you. So like this is an example of an autosomal recessive. And one of the giveaways is that if you're recessive, you know that you have 2 of those alleles. And so if you have a recessive parent, they can only give a little a. Okay, so like this right here, now we don't know who they married, but this person would have the dominant trait, but we would have to know that they were heterozygous because that mom had to have given them a little a. So typically, if you have affected children, there's a good chance that at least one of the parents is affected. Heterozygotes would not have the recessive trait. Notice that affected individuals that are homozygous, they're not going to have children that are affected either, okay? Because it's autosomal, there's not really a significant difference between males and females. All right, some autosomal dominant traits. Remember that to be dominant, you can either be heterozygous or you could be homozygous. And so like in this case here, the reason that this is a question mark is because we really don't know. This girl here could be a capital A, because she could have gotten capitals from her parents, or she could be capital A, lowercase a, and we really don't know what that second one is. Now over here, we know what this one is because that child right there, that daughter, in order for that daughter to be recessive, then this parent had to give a lowercase a as well. And so when you're studying these pedigrees, that's how you can determine the inheritance. As you look at parents, you look at the individual, and then you look at the children, and you can kind of see what alleles they may have gotten. Now, if it's a sex-linked pedigree, there is going to be a difference between males and females. So a couple of things to point out here. Notice all the filled-in squares, okay? Now, there is a filled-in circle, but that's a lot less than 1, 2, 3, 4, 5, like 6 different filled-in squares. Also notice that only circles are carriers. Now, boys can be carriers in an autosomal trait. That's just a heterozygote. But only girls can be carriers if the trait is recessive. So those are some of the things that you can look for in determining inheritance patterns with the pedigree. All right, the last thing we're going to talk about is mutations. And a mutation is any change in DNA. Now, that can happen at the gene level. And a gene mutation is typically just what we call a point mutation. And this is where you have one or more little individual nucleotides that are incorrect. Maybe instead of a C, there's a T. Or instead of an A, there's a G, okay? Now, we're going to talk more about these mutations when we get to protein synthesis. But essentially, I could have a substitution where one letter is substituted for another, an A for a T, for instance. I could have an insertion where one letter is added. And what that does, when we get to protein synthesis, this will make more sense, but it affects the reading of the DNA sequence. Same thing if you have a deletion. If you delete a letter, because we read our DNA and our RNA in groups of 3, if you add one or take out one, it kind of shifts the reading. And so what happens if you have a gene mutation is it could affect the amino acid chain. And that's essentially the protein that's being made. So if we don't have the right order of letters or nitrogen bases, then what can happen is we can affect the protein that's being made. Now sometimes, depending on where it is, there may not be any effect at all. But in general, gene mutations or these point mutations will affect the end result protein. Now, chromosomal mutations, this is where we have like whole pieces or just issues with our chromosomes. Well, this can affect many genes, okay? Not just one, but however many genes are in that section of the chromosome. It could also change the number or the copies of genes. Maybe it gets duplicated. Okay, a duplication is where a piece gets repeated over and over again. And so you can have multiple copies of that gene. That leads to there's a duplication that can lead to a disease called Huntington's disease. A deletion could be where just a whole piece of a chromosome is gone. I think that's what caused the Cri-du-Chat that I mentioned earlier. Inversions is where it gets inverted, like the whole piece gets inverted or flipped. And so what happens is the genetic sequence is going to be reversed. And then a translocation, this kind of looks like crossing over, but what a translocation is where a piece of one chromosome breaks off and attaches to another. So it's not crossing over because that's between homologs. This is between two completely different chromosomes. And so when translocations happen, now one chromosome is missing a piece and one chromosome has an extra piece, and that's going to affect the reading of that genetic code. And so there's just another picture to kind of represent those different mutations. So a gene mutation would ultimately, at most, affect the production of one protein, and then chromosomal mutations could affect multiple proteins because many genes could be affected. Now, sometimes we end up with different numbers of chromosomes, and we actually took a look at this when we studied meiosis, but we could have something called non-disjunction, and that's where the chromosomes are not separated properly in meiosis. I do want you to notice this could happen in meiosis 1, in anaphase 1, or it could happen in meiosis 2, anaphase 2. Either way, it's going to result in gametes that could have an incorrect number, okay? We want to have half, but what happens is you could end up some that have extra and some that have none. And so if these gametes were to be used to produce offspring, then the offspring would have aneuploidy or an abnormal chromosome number. And so here's just a few examples of some disorders that result from non-disjunction. Down syndrome, which is also known as trisomy 21. Now, I don't have a copy of this one, but if a person had Down syndrome, there would be 3 chromosomes here. Okay, once again, that's called a trisomy because there's 3. Turner syndrome is what we call a monosomy. And this is not in the picture either, but this would be a person where there was only just one X. There would be no Y, there would not be a second X. So it's a female that only has one X, okay, instead of XX. Another one is Klinefelter's, and that one's here. You can see that there's 2 X's and a Y. This does not lead to a combination of male and female parts. Okay, this person is a male, but they do have some differences in both inside and on the outside. You wouldn't necessarily know they have it just by looking at them though. A couple others that are not on the list, you can see Edwards here. That's a trisomy 18. They have an extra 18th chromosome. You have Patau syndrome, which is trisomy 13. You can see there's an extra one there. Both of these can be relatively severe. And then there's some that don't exist. Like I've never heard of a trisomy 1 because in some cases, if you were missing a chromosome, that information could be so critical that the baby never completes development. So some of these disorders, they don't even exist. Like I've never heard of an abnormal chromosome number 1 or 2 or 3. And that's just simply because maybe the baby doesn't survive to identify a disorder. And that is going to wrap up our more genetics. This is going to be the last set in this unit. And so we're going to be doing some practice over the next several days. We're going to be doing some more Punnett squares and a karyotype. And then after we finish that material, we're going to wrap it up with a test over genetics. So I hope you guys are doing all right. I do want to encourage you that if at any point you do not understand the Punnett squares, you need to get help, okay? It's something that you need to understand, and once it clicks, it clicks. Some of you guys may have remembered from freshman year, but if not, anytime you need help with something like the Punnett squares, you're going to need to get in during claim time or during Hawk Help and get help from your teachers. As always, feel free to email me with any questions. But this is something that we do have application, and you just want to make sure that you know how to do those problems before we get to test time. And hopefully I will see you guys soon. If you have any issues, just get in touch with me. Y'all have a great day.